The human body is a biological machine. It converts the energy stored in your food into every physical action and passing thought you generate. Yet you experience yourself as a single, unified entity that smoothly senses, thinks, and remembers. The biological reality is that your brain is a highly decentralized network comprised of roughly 86 billion isolated cells called neurons. These billions of cells are physically separated from one another. They do not touch. This presents a clear mechanical problem. How do completely isolated components coordinate to produce seamless movement and consciousness? To understand how the brain operates, we have to trace the mechanics of neural communication. We will start with the internal electrical pulse of a single cell, and then examine the chemical bridge that connects one cell to the next. Everything you perceive or do requires the brain to bridge the microscopic physical gaps between its own cells. An electrical signal conveys information almost instantaneously, but human tissue is a relatively poor conductor of electricity. If your axons conducted electricity the way a copper wire does, the impulses would get weaker and weaker as they traveled across the body. The nervous system solved this signal degradation with a process called the action potential. When an axon is not stimulated, its membrane sits at a resting potential. The inside of the cell holds a negative charge of exactly -70 millivolts relative to the outside, maintained by a high concentration of sodium ions kept outside the cell and potassium ions kept inside. When input from other neurons excites the membrane and pushes that electrical charge up to a threshold of -55 millivolts, specialized gates in the axons suddenly open. Positively charged sodium ions rush into the cell, drawn by the negative charge inside. This influx flips the localized electrical charge from negative to positive. Milliseconds later, the sodium gates snap shut. Potassium gates open, allowing positively charged potassium ions to flow out of the cell, driving the interior back down to its negative resting potential. As the positive charge enters the axon at one point, it triggers the exact same gates to open on the adjacent section of the membrane. The signal propagates in a rapid chain reaction. This sequence is governed by the all or none law. An axon cannot vary the strength or velocity of its action potentials. It is not a dimmer switch. It is a strictly binary system. It fires at absolute full strength, or it does not fire at all. Because the action potential regenerates itself at each point along the membrane, a touch sensation from your toe reaches your brain at the exact same strength it started. The digital pulse perfectly solves the problem of distance, but it hits a literal dead end when it reaches the physical boundary of the cell. When the action potential reaches the very end of the axon, it cannot jump the empty physical space to the next cell. That gap is called the synapse, the specialized microscopic junction where the electrical signal must be translated into a chemical message. This diagram illustrates the process. The unvarying electrical pulse travels down the fiber until it hits the swollen tip of the axon, called the presynaptic terminal bouton. The arrival of the action potential forces the neuron to release chemical molecules, neurotransmitters, into the empty gap. These neurotransmitter molecules diffuse across the neurophysical void, moving toward the membrane of the receiving postsynaptic cell. Once across the cleft, the chemicals dock into specific receptor sites on the receiving neuron. Neurotransmitters bind to receptors based on a specific structural affinity. Only molecules with the correct geometric shape can dock into these protein sites to trigger an excitatory or inhibitory response. Unlike the rigid on or off electrical pulse of the action potential, these chemical messages are highly variable in both strength and duration. A quick, sudden chemical burst handles processing for vision and hearing. Slower, minutes-long chemical messages govern states like hunger and thirst. But to prevent a neuron from firing infinitely, the chemical message must be quickly terminated once the signal is sent. The brain relies on a process called reuptake. The original neuron acts like a vacuum, pulling the released neurotransmitters back up into the cell to clear the synapse. The electrical action potential provides the necessary speed and distance, but the chemical synapse provides the nuance and analog control required for complex behavior. Psychoactive drugs do not create novel brain functions. They strictly manipulate the existing synaptic architecture. They fall into two categories: agonists, which amplify a synaptic signal, and antagonists, which decrease it. Stimulants, specifically cocaine and methylphenidate, provide a clear case study of how a chemical can hijack this system. This diagram shows the junction point. Stimulants physically block the reuptake proteins on the presynaptic membrane, effectively breaking the neuron's vacuum system for the neurotransmitter dopamine. Because the dopamine cannot be cleared away by the blocked reuptake channels, the molecules become trapped in the synaptic cleft. The trapped dopamine molecules continuously bounce off the bottom membrane, repeatedly activating the postsynaptic receptors over and over again. This continuous, unnatural chemical bombardment at the microscopic synapse translates directly to the macroscopic behavioral output of intense arousal, focus, and motivation. There is a biological cost to this hijack. The presynaptic neuron is forced to release dopamine far faster than it can synthesize replacement molecules. Once the drug wears off, the neuron's depleted dopamine reserves result in a prolonged period of lethargy and depression until the supply can be rebuilt. Dramatic, subjective shifts in human consciousness, from manic euphoria to deep depression, are the macroscopic behavioral results of microscopic traffic jams at the synapse. The nervous system relies entirely on this translation, an unvarying digital electrical signal within the cell, handed off to a highly variable analog chemical signal between cells. Whether it is L-DOPA restoring smooth movement in Parkinson's patients, or hallucinogenic compounds drastically distorting a person's visual reality, all external chemicals are bound by this same structural logic. They must physically bind to receptors to alter the behavior of the cell. Now, we scale this cellular junction back up to the entire human brain. No single action potential and no single flooded synapse is capable of dictating a complex human behavior on its own. Our continuous, subjective stream of conscious experience exists because trillions of these microscopic electrochemical handshakes are happening simultaneously. The translation of an electrical pulse into a chemical signal allows these billions of isolated cells to operate as a single coordinated system.